Hello, everyone. Welcome to this week's edition of Monday Minutes. This week in lecture, we're moving on from supporting connective tissue, including bone, cartilage, and joints, and we're beginning our discussion of the third kind of tissue found in the human body, which is muscle. Over the next two lectures, we'll be talking about the three different types of muscle tissue, including skeletal muscle, cardiac muscle, and smooth muscle. We'll be spending most of our time discussing skeletal muscle. Remember, skeletal muscle is just a tissue, and all tissues are composed of cells and extracellular matrix. Every type of tissue has its own list of functions, including skeletal muscle. Some of those functions seem obvious to us. We know that skeletal muscle, for example, helps our body move. We also know that it helps us stay upright and keep our normal posture, along with the bony skeleton. The things that might not be as obvious to us are things like the fact that skeletal muscle helps protect our internal organs. Skeletal muscle is attached to our bony skeleton, and those two things together help protect the structures that are deeper inside of our body, all of our various internal organs. The other two things that skeletal muscle does for us, though, are helping to maintain our body temperature and helping blood and lymph fluids flow through the circulatory systems. We've talked about the complex process that maintains our core body temperature within its normal range. One of the effectors that we discussed was skeletal muscle, and we said that it's the contraction of skeletal muscle that can help our body warm up when our core body temperature starts to go down within the normal range. By contracting, skeletal muscle actually generates heat, and it's going to help our body temperature start to move up within its normal range. In terms of our circulatory systems, remember you have two. You have one system that pumps blood, and you have another system that contains lymphatic fluid. The blood circulatory system has a pump within it, your heart. The lymphatic system doesn't, though. And the lymphatic system relies completely upon our physical movement through our environment to push those lymphatic fluids through our body. When people get very sedentary and they stop moving from time to time through the day, lymphatic fluid can start to pool in tissue, and we can develop abnormal swellings called edema and lymphedema. Skeletal muscle also helps our blood circulate, particularly on the venous side. The contraction and relaxation of skeletal muscles and the movement of the bony skeleton helps push blood through the venous side and back to the heart. In the lecture, we talk quite a bit about the structure of skeletal muscle tissue, and we spent a good amount of time discussing the cells themselves. Skeletal muscle cells are called muscle fibers, and for a good reason. They are shaped very differently from the other cells that we have seen so far. They don't look like epithelial cells. They don't look like fibroblasts or osteocytes or chondrocytes. They look elongated. They have a very elongated fibrous shape, which is why they're called muscle fibers. The other thing that makes these cells particularly unusual is that they are multinucleated. We talk in the lecture about how skeletal muscle fibers are formed during fetal development. It involves the coming together of several individual cells to create the one skeletal muscle fiber. and that's why it has multiple nuclei. There's a lot of anatomy that goes along with this new kind of cell, this skeletal muscle fiber. You can see on this slide that a lot of the terminology begins with the term sarco, the sarcolemma, the sarcoplasmic reticulum, sarcomeres, and so on. Anytime you see the term sarco, think muscle. The other thing that makes skeletal muscle fibers quite unique is the presence of a protein called myoglobin. Myoglobin is a relative of the protein called hemoglobin, which is responsible for carrying the gases oxygen and carbon dioxide through our blood and to and from our cells. Myoglobin is a very similar protein in that it's capable of binding oxygen. So inside of a muscle fiber, the myoglobin holds onto extra supplies of oxygen for when they're needed in the production of ATP. In the lecture, we talked about how myoglobin should never be present in the blood the way hemoglobin is. The only time we will find myoglobin in the blood is if muscle cells have been damaged in large numbers. There are lots of really good illustrations and light microscope images for you to use in this lecture, like what you see on this slide. Up at the top, the artist shows us a representation of a skeletal muscle fiber, showing us that this cell is unlike any other cell that we've seen so far in the course. It's elongated, almost like a cigar. It's also striated or striped. Inside the cell, in addition to all of the organelles that we would normally find in a human cell, we find much of the space taken up by these elongated structures called myofibrils. Down at the bottom in the light microscope image, we can also see these striations present in the elongated muscle fibers, as well as the multinucleated appearance. Muscle tissue is highly organized. In skeletal muscle tissue, we can describe many layers of organization from the large, grossly visible muscle itself, all the way down to the individual myofibrils that are found within a single muscle fiber. The diagram also shows us where certain anatomical structures are wrapped in a fibrous covering. If we go from the large, grossly visible muscle, you can see that there's a fibrous covering around it called the epimysium. Remember, the epimysium, this outer covering, is actually not the outermost level of tissue that you'll find around a muscle. There's also a very heavy layer of connective tissue called fascia, which occurs outside of the epimysium. If we go inside, we pull the epimysium off, what we're going to find are individual bundles called fascicles. And these fascicles are each wrapped in their own fibrous covering called perimysium. If you took one fascicle and looked inside, you would find many, many individual muscle fibers, individual cells. Each cell is also wrapped in a layer of fibrous tissue called endomysium. If we took one muscle fiber out, like we've done here, you can see that it's multinucleated, and you can see it is full not only of ordinary organelles, but of these elongated structures, again called myofibrils. The other thing the artist has drawn here are these little purple structures. These represent mitochondria. Remember, muscle tissue, particularly skeletal muscle, has to have a lot of mitochondria in it because it has a very high demand for ATP. This slide, too, shows a drawing of these levels of organization within skeletal muscle tissue, from the grossly visible muscle, something like a biceps brachii muscle. We can pull out bundles of individual cells called fascicles. And if we take one cell, and that's this structure right here, one cell, one muscle fiber, we can see that it's striated. Inside that cell, we will find all of the organelles that we find in any human cell. But in addition, we find the cytoplasm just packed with these long structures called myofibrils. Each myofibril is also striped or striated. The artist has pulled out one myofibril here and shown us that along the length of the myofibril we find individual sarcomeres. And remember, the sarcomere is the unit of contraction within skeletal muscle. This is another drawing that you should be sure that you understand in all of its complexity. It represents an individual muscle fiber that has had its plasma membrane removed, or at least cut open. Remember, in muscle fibers, the plasma membrane gets a special name. It's called a sarcolemma. If we pull off most of the sarcolemma, we can see all those elongated myofibrils inside the muscle fiber. We can also see lots of mitochondria. The other thing that we can see that looks a little unusual is this blue sort of netting material that surrounds the myofibrils. It not only surrounds all of them on the outside, but it dives in between and around each individual myofibril. This netted or mesh structure is called the sarcoplasmic reticulum. It's an organelle that is only found in muscle. The other thing to notice is that the sarcolemma, again, this yellow membrane, folds inward at certain lengths. It folds inward and creates what are called T-tubules. The T-tubules are nothing mysterious. They're just infolded areas of the cell's membrane, or the sarcolemma, and they serve a very important function during contraction. You'll notice that each T-tubule is surrounded by two blue tubules that represent terminal cisterna. The terminal cisterna are just parts of this sarcoplasmic reticulum. So one T-tubule surrounded by two terminal cisterna makes up a triad. And a triad is functionally important during contraction. The other thing to notice in this drawing is that you can see the striations along the length of the myofibrils. Remember, as we travel down one myofibril, we're going to see one sarcomere followed by another, followed by another, followed by another. That's what myofibrils are. They're collections of filaments, thick filaments and thin filaments that are very precisely organized around each other to form these units of contraction called sarcomeres. So once we start discussing sarcomeres in the lecture, we're now talking about microscopic anatomy. The sarcomeres that are inside the myofibrils in each muscle fiber are made of proteins, two primary proteins and several accessory proteins. The primary proteins are myosin and actin. The myosin proteins make up what are called the thick filaments, and the actin makes up the thin filaments. In addition to actin and myosin, there are many other what we call accessory proteins that help the individual sarcomeres keep their shape. It's critically important that the thin and thick filaments are arranged around each other in perfect organization. If they're not arranged that way, if these accessory proteins were not present, and the thick and thin filaments were not correctly arranged, the muscle would not be able to contract. In the lecture, we spend a lot of time talking about sarcomeres in 2 dimensions, but it's very important that we always remember that inside an individual myofibril, which is what this represents, the sarcomeres are actually arranged in 3 dimensions. In fact, if you look at this cross section, this transverse section through a myofibril, you'll see this. You'll see these larger circles surrounded by these smaller dots in a hexagonal pattern. The larger circles represent myosin, particularly the globular heads in cross-section, and the smaller dark dots represent the thin filaments that surround each thick filament. So, remember that while we often think of sarcomeres in 2 dimensions to imagine how they shorten during contraction, within the myofibril they're arranged in 3 dimensions. Remember that an individual sarcomere runs from one Z-line to the next Z-line down the length of a myofibril. Underneath the microscope, a sarcomere looks like this. Here's one Z-line, here's the next Z-line, so everything between them is one sarcomere. We describe bands and lines within the sarcomere. Everywhere you see this darker coloration from here all the way over to here is referred to as the A band. On each side of the A band, where it's more lightly colored, these are called the I bands. Now let's think about what makes these bands. The A bands are formed where the thick filaments and the thin filaments overlap each other. That's what gives it its darker color. In comparison, the I bands are formed where only thin filaments overlap each other, and that's what gives it the lighter color. In addition to the A band and the I band, we can also see a line through the very center of the A band. This is called the M line, and this is where the myosin proteins are bound to each other in mirror symmetry. There's a bundle of myosin protein over here, and there's a bundle of myosin protein over here, and the 2 bundles are joined together at the M line. The other thing we know is that there's a light band within the center of the A band. This is called the H zone. This is the place where we have myosin thick filaments overlapping each other, but this is the place where there are no heads present. There are no globular heads present in the H zone. Remember, these myosin heads come off of the thick filaments in 360 degrees, and they're staggered. It looks sort of random to us, but the truth is the myosin heads are arranged symmetrically around the 360 degrees of the thick filament. It's the myosin heads that are going to do most of the work of contraction. The myosin head is just a region of the myosin protein. The other thing to know about the myosin head is that this is where there's another region called myosin ATPase. In other words, part of the myosin head acts like an enzyme, and it's able to split ATP. So one individual myosin protein looks like this. It has an elongated region and then these 2 globular heads coming off. And within those globular heads, we have myosin ATPase activity. Myosin ATPase breaks ATP into ADP plus a phosphate group plus energy. The thin filaments in the sarcomere also have their own critically important anatomy. Remember, a thin filament is made out of 2 strands of actin molecules that are all bound together. F-actin is the name we give to actin molecules that are bound together in a string or a filament. G-actin is the name we give to an individual molecule of actin, or globular actin. So we have 2 strands of F-actin, all bound together and wrapped around each other like a helix. That's the actin portion of the thin filament. But you'll notice that there is other protein associated with the thin filament, and these are regulatory proteins. The green protein that is elongated and looks a little bit like a licorice stick is tropomyosin. The much smaller yellow and orange protein is troponin. Troponin has 3 separate areas within it. It has TNI, which is the portion that binds to actin, TNT, which is the portion that binds to the green tropomyosin molecule, and TNC, which is the portion that's capable of binding to calcium. Remember, calcium plays an important role in regulating the sliding of the sarcomere, which allows contraction to occur. The last thing we discussed in the lecture is the fact that there are actually different types of muscle fibers. There are 3 different types that we can describe. So if we take a muscle, such as the biceps, once again, and we look inside that grossly visible muscle, we're going to find some mixture of these 3 types of muscle fibers. Each type of muscle fiber has a different type of contraction and means of supplying ATP. Remember, when we talk about type of contraction, we're talking about power and we're talking about speed. The power of a contraction within a muscle fiber is related to the diameter of that fiber. Some muscle fibers have a smaller diameter and some have a larger diameter. The larger the diameter of a muscle fiber, the more myofibrils it has within it. And the more myofibrils it has, the more sarcomeres it has. The more sarcomeres that are in there, the more powerful the contraction will be. The other component of type of contraction is speed of contraction. We discussed 2 speeds, what we call fast twitch and slow twitch. So some muscle fibers, the ones that have a larger diameter, are fast twitch type. That means the initiation of the contraction is faster in those types of muscle fibers. And it has to do with a particular variation that they have in that myosin ATPase area on the myosin heads. These types, these fast twitch types of muscle fibers, are going to produce a contraction of shorter duration. So what you gain in speed, you lose in duration. Slow twitch muscle fibers are very different. These are the ones that tend to have a smaller diameter. They're going to initiate contraction more slowly, but the contraction that they produce is of longer duration. The other way that we classify muscle fibers is according to how they get their ATP. And here there are 2 different metabolic pathways that will produce ATP in the muscle fiber, either aerobic cellular respiration or just glycolysis. So the muscle fibers that are using aerobic cellular respiration are making their ATP very efficiently, but relatively slowly. We refer to these as oxidative fibers or red fibers. That's the other term you'll hear. Oxidative fibers have to have an extensive blood supply to them. They have lots of myoglobin in them because they need that extra store of oxygen to do cellular respiration. They have a large number of mitochondria in them because that's where cellular respiration takes place. And they tend to be resistant to fatigue. In comparison, there are other types of muscle fibers that use glycolysis to make their ATP. This is an inefficient way to make ATP, but it's relatively fast. Glycolytic fibers, also called white fibers, need less blood, so they tend to have less blood supply to them. They will have less myoglobin in them because they don't need oxygen to perform glycolysis. You'll find fewer mitochondria in these types of muscle fibers, and they do tend to fatigue more quickly. When you put all of that together, when you put together the power and the speed and the type of metabolic reaction that the muscle fiber uses to get its ATP, we can put together 3 types of muscle fibers, and we have them listed on this slide. Remember, we call them slow oxidative, fast oxidative, or fast glycolytic. Slow and fast, that refers to the twitch speed. Oxidative and glycolytic refers to the type of metabolism that muscle fiber is using to make its ATP. The other thing we discussed in lecture is that, for example, slow oxidative fibers are also sometimes called type 1 fibers, and the other ones are called type 2A and type 2B or type 2X fibers, but we're not using that language in our course. We're going to use the language shown in red. Slow oxidative or SO fibers, fast oxidative or FO fibers, and fast glycolytic or FG fibers. Remember, in muscles, you're going to find some combination of these fibers in different proportions or different percentages. It depends on the work that particular muscle has to do. There are some muscle types, remember, that have to work very quickly. They need to be able to perform a very swift but brief contraction. Things like your eye muscles or your hand muscles. You're going to find a high percentage of fast glycolytic fibers in there. In comparison, the kinds of muscles that help us maintain our posture, things like your back muscles or the muscles in the calves of your legs, they need a type of contraction that is much more slower and much more longer lasting. So those types of muscles are going to be high in slow oxidative fibers. It depends on the muscle. It depends on the work that muscle has to do. That determines the percentages of SO, FO, and FG fibers. Remember, too, this information at the bottom of the slide. What determines the percentages of each muscle fiber type is partially due to how fit a person is and partially due to genetics. We talked in the lecture about how when we get to very high levels of athletic competition, everything comes down to the genetic lottery. At Olympic levels of competition, there are some people who are just born with the right concentration, the right percentage of muscle fiber types for their chosen sport. There are other people that may desperately want to be at the Olympic level, and they train very, very hard to get there, but they never quite get there. And that's because they simply were not born with the percentage or proportion of muscle fiber types that is needed to perform at that high of a level. That brings us to the end of this week's Monday Minutes. As you're watching the lecture and taking notes, don't forget to pull out the topic concentration guide for the next lecture exam and add your own notes into the guide. Remember, the topic concentration guide is there for you to create your own customized study guide by adding in additional information from your lecture notes. The last thing I wanted to mention in today's Monday Minutes is something about our discussion boards. Remember, in one of our discussion boards, I post practice essay questions for you so you can get some practice writing out essays related to the lecture topics for the next exam. There's a handful of students in class who have been taking advantage of these practice essay questions. Good for you. But most of you have not. It really is a good experience for you to look at these practice questions and write out an essay, because it will allow you to start thinking about the kinds of essay questions that just might show up on the next exam. Remember, too, that there will be an essay question on the final common assessment. So the more practice that you can get writing essays about the complex physiology that we're discussing, the better the chances of you earning more points when you have to do it for real on the exams. Okay, everyone, enjoy the rest of your week. If you have any questions about this week's lecture material, send me a message through Canvas.